Okay, so let's go ahead and actually get started now. So today's workshop is going to be a little bit different because we're actually going to be doing an activity together. And Chris, who is the postdoc in Dr. Chen's lab, is going to be walking you through how to actually use these virtual machines that we've set up and hopefully everyone's been able to connect to in order to actually process the raw sequencing data. Because if you remember how we left it last time, there's some data sets that you actually have to go all the way back to the raw data in order to process. And this is how you would actually go about that. So without further ado, Chris, why don't you go ahead and get started?
Yeah, thank you, Henry. So just before I start, just want to make sure everyone has the access to the virtual machine, right? Already logged in? Oops, I couldn't see the chat box. Supposed to be fine for everyone?
Yeah, if anyone's having any issues, do please put that in the chat. It's very important that we all get connected to these. Oh, also, I completely forgot to mention, I'm going to put the link to the box folder for this lecture. I'm going to put that link in the chat in just a minute.
Okay. Yeah. So today I'll be talking about the RNA-Seq mapping and the risk quantification. So mostly almost 80% using the Linux. And yeah, okay, so the first, let's talk about just very briefly talk about the RNA sequencing. So this is a typical pipeline or workflow for Illumina-based sequencing. So we have the RNA, your RNA samples, and convert it back to the cDNA. Here's for the DNA sequencing, but it's very similar for RNA sequencing. So we convert everything to the cDNA, and then authenticate or fragment the cDNAs to small fragments. So it's typically a few hundred bases. And then we add adapters and prepare the libraries for sequencing. And after that's good to be loaded to the flow cells and being sequenced. So the sequencer basically try to read the ATCG, so the sequence of each fragment of our sample. So the results, the output of the sequencing will be a bunch of sequences. So you see that like ATCG. I'll give you a view of the results in the next slide. So it's basically a bunch of sequences and with some stats or some quality values associated with the sequencing results. So here's a snapshot of a paired sequencing. So what I mean, I'm kind of blocked by the video. Okay, so what I mean by pair end is suppose we have the DNA fragment, cDNA fragment we want to read the sequence. So there are two ways to read the sequence. So the first one is the pair end. So we read both ends of the DNA fragment. And the other one is single end. Single end, we only read read the sequence from one end. So we don't have the second read. So here's an example of the pair end sequencing. So if you have a pair end sequencing, the raw data you will have is the FASTQ file, and it typically comes with R1 and R2. So it's a pair of FASTQ file for each of your sample. So R1 means it's read one, so it's from the left-hand side, and R2 is from the right-hand side. So each of so you see the FASTQ. So I'll show you how to visualize, how to read the FASTQ in Linux in a bit. But if you view the FASTQ file in Linux, you'll see every read covers, every 4 lines represent a read. So the first line is an identifier of each read. So you see it's a bunch of numbers. So I think it has the ID of the flow cell and like the location of the specific read from the flow cell. So you have a bunch of numbers there. So one means it's read one, and you see from the read two, you have the same ID, but it starts with a 2. So which means it's your R2. And the second line right here is the sequence you have. So that's the exact sequence of the first sequencing read. And the third line is always a plus sign. And the fourth line, see a bunch of characters and numbers and even symbols. So those represent the sequencing quality of each read. So the first symbol here represent the quality, the sequencing quality for the first base. So it's a base by base quality score. So it's a ASCII, engineered way to represent scores. But scores are numbers. But they're just trying to present the numbers by characters and by symbols. So those 4 lines correspond to the first read, and the second 4 lines are the other second read. So that's the raw data you get from typical sequencing run. And the format are the same, no matter you are running like DNA-Seq, RNA-Seq, the format are exactly the same, but the contents may be different. The contents here represent the cDNA. If you have the DNA samples, the contents represent the DNA. Okay. Oops. Okay. So I also want to talk about the genome view of the genes. So we have, if you line up all the chromosomes, we have all the way from chromosome 1 to chromosome X and Y. And here are the number of genes, the gene distribution across all chromosomes. So if you zoom in to specific chromosome, I just randomly chose one. So if you zoom in to a specific cytoband, you see multiple genes are located in a cytoband. If you further zoom in to a gene, so just randomly chose the KMT2E. So if you zoom into a gene, so you have the introns and exons of the gene. And further zoom in, you'll see the ATCG, the DNA sequences of the exon. And you also have the amino acid coded with the codons associated with the DNA sequences. So the key computational challenge of sequencing data is how you link those sequences back to the genome or back to the gene. Because when we sequence when we get the sequencing data, it's a bunch of the sequences. So for RNA, for typical RNA sequencing, we have roughly 20 to 30 million of sequencing reads. So you obtain 20 to 30 million of those small fragments, small reads, small DNA sequences. So we don't know where those sequences come from, and there's no way we can count it before we do alignment. So that's why the key computation idea here is to align those sequences back to the genome or back to the gene. So to identify where those sequences come from. So if you know this sequence come from this KMT2E gene, then you can count it as expression 1. So if you have like 10 reads aligned to the same gene, then expression level will be 10. So that's the key rationale of the RNA sequence analysis. So there are mainly two workflows for RNA sequence analysis. We'll be focusing on the second one today, but the first one is actually the alignment based. So we have the sequencing reads, that's the raw data, the ATCG reads. So I store in the FASTQ file. So we typically do the simple quality control, which is not covered today, but it's simple. So after the quality control, and then we align all the reads, each of the reads back to the genome. Whether depends on your pipeline. Sometimes we align it back to the entire genome, which is 3 times 10 to the 9 bases, 3 billion bases, the entire genome. Or sometimes we only map to the transcriptome, which account for 2 to 3% of the entire genome. So it depends on the alignment or the experiment design. So we align it back to the genome, find where those sequence reads come from, and then we do the counts. So here's the illustration. So if you do the alignment, so that's the typical output of the alignment. That's the band file. So right here, so band files. So the band files contain the same. So you have the ID for the sequencing read. So this ID will be the same as the one you have in the FASTQ file in the raw data, and this is the sequence you have for the sequencing read. So those information are the same from as appear in the FASTQ file in the raw data. And the only difference is that after you do the alignment, band file contains the chromosome location, the first chromosome and the location. And it also contains the 101N, means it's 101 matches. The entire sequence. So each sequence is of length 101, and those 101 are perfectly matching the DNA sequence of this location. So we can say that this sequence comes from that location, that specific location. So after you do all the alignment for all of the 20, 30 million reads, then you have, everything is contained in the band file, so you can visualize those band file by some software, and it's called IGV if you are interested. So so with the visualization tool, you can see, given a specific gene, we have multiple alignments. So each line here is a read. So we have multiple reads aligned to this gene. So the typical way to count the gene expression is that for this gene, so it's multiple gene, kind of merged together. But the way to count the gene expression is that we count the number of reads that fall into this specific gene. So we can count, like, say, if it's like 1,000 reads here, then the gene is expressing at 1,000 reads. So that's the way to quantify gene expression by the alignment-based method, so the left-hand side. And today we're talking about, oh, by the way, so left-hand side, because it requires alignment. So it could be much, much slower than the methods we are introducing today. So typically, those tools, for each sample, it takes a couple of hours, few hours for on a high-performance computing server. So it takes some time, but it aligns everything back to the genome. So it provides you the location, the chromosome location of your sequencing data. And the pipeline we're introducing today is mostly on Salmon. So it's a alignment-free pipeline. Somebody call it like pseudo alignment. So basically means it doesn't involve the alignment process. So it doesn't really map the reads back to the genome. So here's how it works. Okay, so they call it pseudo alignment or like quasi mapping. There are like several ways to describe it. So that's the figure I extract from their website. So Salmon, they say don't count, quantify. So by counting, it means the traditional way. So we align everything back to the genes, and then we count how many reads fall in each gene. So that's the counting method. And the Salmon, they say don't count, just simply quantify. So how they do is I'm just doing the conceptual introduction today. So how they do is we have the raw reads, right? We have all the sequences we obtain from the RNA sequencing. That's our samples. And in the human genome, we know there are, all the transcripts and their sequences. So we know pretty much all the information. So Salmon pre-computes the index. So that's the step called indexing. So they compute pre-compute the indexing by finding those representative or those unique sequences that only appear in some of the transcripts. So if you see the small, specific sequences in your read, say the read one contains specific sequence, and that sequence only appear in one of the transcript of the human genome, then you know this read belongs to that transcript. So by doing so, we are not really aligning these reads back to the 3 billion bases to find where it locates. So we are basically comparing the sequence and try to associate the reads, the sequencing reads, back to the transcripts they represent. So the transcripts are small. So it's much shorter than the entire genome. So the time, the efficiency of the Salmon can be very, very high. So it basically compares our samples to the Salmon index and do the mapping. I'm not going to the too deep here. So that's the way Salmon works. Okay, so I'll briefly, I can probably explain these codes first, and then we can go to the virtual machine. So today we're gonna do the prefetch. So we'll download the sequencing raw data from the SRA. So I'll show you the website after this slide. So we'll download the SRA data from, we'll download the sequencing data from the SRA. So this is the specific sample we'll be downloading. So after we download the data, we'll prepare the FASTQ file. So FASTQ is the file that contains all the ATCG, all the sequences, and then we'll run the Salmon quantification mode. So it's basically 3 main lines. Three functions we're gonna run today. So after this, we'll have the output results. So I'll walk you through each step later. Okay, so let's take a look at the SRA first. Any questions so far?
Simon asked a really good question, which was, what's the advantage of doing the alignment versus salmon? And he said, is there a reason that we would ever use the more computationally expensive one?
Yeah. Right. Good question. Good question. So salmon, I would say the only application of salmon is to quantify, to generate the abundance table. So if your interest is only on the expression level of your samples of each gene, each transcript, then salmon is a good option. So I have like a few more slides. When we do the hands-on session, after we do the hands-on session, I have a few slides to quickly introduce other alignments, or like how other like huge consortiums do alignments. But traditional alignment takes time, but traditional alignment provides much more information, like the chromosome locations. And if you want to do like fusion, if you want to call mutations from the RNA sequencing, some people do mutation calling from RNA sequencing. So by using salmon, you couldn't call mutations. But if you align it back to the reference genome, and then you see like a single base, your reads are different from the reference, then you know there's a potential mutation there. So there are benefits for traditional alignment-based methods. It really enable many other functions beyond quantification. But if your goal is to simply to quantify the gene expression profiles in your data, in your RNA-seq data, then salmon is a good choice because it's really fast. Yeah. Okay. So here's the GEO. I think Henry introduced the GEO a couple weeks ago. So GEO, you can search for the specific gene expression data set here. So just type in the keyword like a specific disease or a specific cell lines. You can type in here. Then you can search for all the data set associated with, all the published data set. So GEO contains both the microarray, the traditional way of gene expression, the SNP arrays, everything, microarrays and the sequencing. So GEO contains those. So here's the page for of GEO on the airway data set we're using today. So it contains the descriptions of the data processing, the study design, and I probably need to go back. Yeah, so that's the entire data set, the airway data set. And the data set contains 16 samples right here. So you see the 16 samples, they have the treated and they have untreated, which is control cells. They also have the treated with the dexamethasone samples. So you see a different treatment there. So if you are interested in specific sample, you can click in there and you can see the specific description of that sample. You see the cell line, the treatments are applied to the sample. And today we're showing by the way, you can, from the GEO, you can download the pre-processed data from right here. You have the pre-processed data, and also from the series metric, you can download the tabulated process data. Those data are typically normalized and pre-processed by the authors. So it typically, different studies do very different ways of analysis in terms of the pipelines or post-processing normalization, like a differential expression. So the data set you download from the GEO are mostly post-processed. Those are normalized data. So if you want to download the FASTQ, the raw data, and really run your like Salmon yourself, then you need to go to the SRA. So from the samples right here, from each sample, you can see their SRA ID. So you can directly search the SRA ID from here, or you can go to the SRA right here. So you just type in SRA and Google it. So there's a database called SRA hosted by the NCBI. So if we, today we'll be using the sample. Let me copy the sample name. Today we'll be analyzing this sample. So if you search, then you'll find that specific sample. So it contains pretty similar information as those on the GEO, and you can also find the experiments associated with this sample. Yeah, so it shows the experiment. The experiment pretty much is the study. So if you click on the all runs right here, so then you see all the samples within this this data set. So you see 16 samples and treated with different drugs and the control samples. So those are the SRR. SRR means the IDs in the SRA. So you have the raw data. So we're trying to download the 521 right here. So today we'll be downloading the 521 and also using Selman to analyze this sample. Okay. Okay, so let's go back to the virtual machine first. I can show you. So here's the virtual machine. So after you log in, you can always do the LL. LL lists all the files, all the directory within your account. So everyone with me? Okay, so here we have already some folders. So those are the output folders Henry already put in there. So we'll come back to this later. So before we start with the functions, I want to introduce you the paths. So we have so in Linux, everything is with command line. So if you want to change your directory, say you see there's a folder called SRR, something right here. If you want to go into the directory and see what's inside, you can do CD change directory and just paste type in the specific directory name you want to enter. Then you are in that directory. So you can do the LL again. You can see the contents within the folder. So directory are pretty much folder in the language of Windows or Mac.
Hey, Chris, do you think you could zoom in just a little bit?
Yeah. Okay. I can do. How's that?
Maybe just one tick more. Yeah, that looks great.
This one. Okay. Yeah. Right. So if you yeah, so that's how we change directory. So if you want to go back to the original directory, you can do cd dot dot. Cd dot means go back to the previous directory. What I mean previous is you can type in the pwd. So it tells you where you are right now. So the folder we are looking at, we are opening right now is this folder. So it's a home, and this is our login. I think it's our account name, and that's the folder we are in. So if you type in cd dot dot, and then you do pwd, then you see we are at the previous directory. Okay, so we use data to go back to the previous upper directory, and if you want to enter a specific directory, you can see which directory you want to enter, and then cd to the directory. So there's how we change directory. And if you want to look at the contents of say like the SR, but you don't want to enter it, there's another way. So we can LL, and we can type in the SRR. So we are not changing the directory. We're not doing CD. We're still at our like home directory. So we're still right here. But if you do the LL and this folder, so you pretty much list all the contents within it, and we are still in our original directory. We are not entering this. So it's sort of a preview. I would say preview mode. So you can look at the contents. And if you want to look at this one, so you can do the LL and within it. So it's a second directory. So you can list all the contents within the folders. So it allows you to do the multiple. So you can do the combination. So you just type in the directory, the path all the way to the folder you want to take a look. So just I can do LL to list all the files. Okay. Yeah. Okay, so that's the change of directory. And if you want to look at the contents of the directories, so the first step we're gonna do today is to prefetch. So prefetch is to download the data from the SRA. So we can copy. I can paste it to the Zoom window maybe. Let's see. Okay.
Just for a little bit of clarification, we put the Prefetch program on your virtual machines. You won't initially have that if you just open up a new computer. So that's something you have to download and install.
Yeah, all the functions we are using today, the prefetch and the fastq dump, the fastq prepare, and also the element are, I mean, you need to install before you want to use it on your own machine, right? Okay, so I've already pasted a function in our chat box. So if you paste the function and hit enter, then it downloads the locally. Oh, I think it's already there, right? It's in the NCBI. We probably already downloaded.
Right. Yeah. Oh, I totally forgot about that. So if you want to delete the
Yeah.
NCBI folder or delete the other folder, and then it should work.
Okay, so let's do this. So if you want to delete a directory, so you just do the rm, it's remove. And if you're removing a file, you simply type in remove and the file name. But if you want to remove the directory, then you need to do rm-r. -r means recursive because it's a directory. Directory contains, you won't know like what it contains. You could contain like directory after directory after directory. So by doing the recursive, it removes the entire directory and everything within the directory. So let's remove the .ncbi. Okay, should be fine. Let's see. Okay, so everyone with me now? So after we remove, you can do ll again, then the .ncbi disappear. And there's no trash bin on Linux, so be careful of the rm function. So I'm not sure whether there's a way to rescue the files you already removed. I personally didn't succeed at all. So be careful of what you remove. Okay, so after that, you can type in the function I just copied to the chat box, the prefetch. This should work now. Oh, I probably shouldn't delete the NCBI. It contains the configuration, right?
Oh, yeah.
Oh, okay.
So, all right. No, this is good. This is good because this is something that's actually
Yeah.
likely to happen to people at some
Yeah,
point.
right. Okay, so we can try to initiate it. I think it's easy, right?
Yeah, well, let's back up. So it's got a good warning message. The warning message tells you before continuing, please run this thing. So this is why it's so good to read the error messages you get.
Yeah. So it shows the remember we already pre-installed the what's it called the prefetch for you. So if you install the prefetch first time and you want to use it for the first time, then you need to do the configuration. So this is how you do the configuration. So you just simply copy this one and just type in. I think everyone already deleted the .ncbi, right? It's probably too late. And we don't need to do anything actually. So just do, you just hit S here. It's like stands for save. Okay. And then exit. I think that will do it. Everyone with me? So do the S, small s, and it's safe. Just hit OK. And then do the X right here. So your exit. Yeah, let's see. Yeah, we have, yay, it's back. So the .ncbi is back. So I think it's back with all the configurations we need. Let's try again. Okay.
Yeah, so I think you have to delete the other folder.
Which one? The SRR?
Yeah, the SRR one.
Yeah, or we can try like another just SRA number maybe. Just a demonstration.
If you delete that SRR folder, it'll work.
Okay, remove the SRR. Okay, everyone with me now? It's kind of sorry for the confusion. So now we have we already removed the SRR. So the SRR was we pre-downloaded. So just in case we couldn't run the prerequisite the prefetch, but ends up I mean we already download the file and the prefetch doesn't work because it's already exists. Okay, let's download again. Yeah, now it works. So this step, this function downloads the data directly from the SRA. It doesn't take long. I think it's only like few minutes, very short time. So you'll download the entire SRA file to your local machine. So now we are downloading to the virtual machine.
So I have a quick question. So if we were doing this on our own computer that hasn't had any of the stuff preloaded, so we would need to upload the .ncbi file, the salmon .out, and what other files and the SRA and the SRR file?
So would you say again? So are you mean like for the first time, if you want to use the software for the first time, what would you need to prepare?
Right. Yeah. Like what exactly is, I just want to know like what exactly is preloaded right now that we would want on our own computer.
Yeah. So if you want to download the data, so pretty much all the functions. So right here, so that's the typical Linux function. So this is a function. So this is a function and Salmon is a function. So every function you want to use, it's as long as it's not pre-installed in Linux, those are not. So all the functions we need to install ourselves if you want to run it on your machine. But if you only need to install for once. So if you install Prefetch and then you'll be there. So you can use it afterwards.
Okay, thank you.
Yeah.
I'll very briefly mention the way that I set up these virtual machines is I use something called Anaconda. Or as more specifically, I used something called Mini Conda. So Conda is a package management system that can deal with all sorts of kinds of packages. So basically, if you just type, if you've got Conda on your machine and then you type Conda install and then the name of something you want to install, it will install it. So that's how I installed all of these packages, with just doing Conda install and then the name of like Prefetch or Conda install salmon. So that's how you can do it if you've got Conda on your machine, which is how I recommend everyone to do things for the most part.
Okay, so we have now downloaded the SRR. So you see oh, by the way, I forgot to explain the code. So the code we just ran was prefetch to download. So that's the function name. So we want to download this sample from the SRA. And we indicate this is a parameter. So it's a typical way Linux functions take parameters. So it's dash parameter name and then your setting. And another way I'll be showing in the Salmon. So another way, if they have a sort of shortcut, so it'll be single dash. So it's single dash or double dash, depends on the software you use. So if you refer to the manual, then you'll see the parameters you have and all the options, all the settings you can have, all the parameters you can have. So we're downloading this sample and we only indicate where our output directory is. So we want to download the sample to this folder. So that's why after we run this, if you do the LL again, you'll see we have a new folder. The time is kind of weird. It's probably at the Europe somewhere. Yeah, so now we have this folder, and if you look at, so we can cd to the folder test. Just a trick you can, type, when you type in partially the directory name, you can hit tab. So if it's unique, then it auto fill the entire folder name for you. So you can hit tab to finish. And then you do the LL again. So you have we already downloaded this sample, so you can cd to this directory. Then list again, you see there's a SRA file. So I'm not sure whether you can visualize the SRA file because it's a NCBI SRA format. So the next step we'll be doing is to convert or to extract the FASTQ from the SRA. Okay. Okay, so let's directly copy. Okay, so the second line will be running is this one. I'll paste to the chat box right here. Okay. So remember now we are at this directory. So you can hit PDX. So now it's where we sit right now. And the function, the code I just paste in the chat box is actually relative to our home directory. What I mean is let's change back to the home directory. So if you are going to the back to the home directory, there are two ways. You can type in this, the home and your account name, or you can simply type cd tilde, then it goes to the home. So tilde stands for home, your home directory. Okay, so if you hit pwd again, so now we're right here, and the contents are this. So the function I just put in the chat box is right here. So let me explain it. So this is the function to dump FASTQ, so to extract and to write the FASTQ. And -e means how many threads we are using, how many computing nodes we are using. So we're using 14. So I think each of your virtual machine has 16 threads. So we're using 14 of them. So typically the more threads you use, the faster your job is. But it's really not linear. So for some of the like computation heavy task, especially when you like read and write disk very often. So if you really hit a huge number here, it's not gonna, the performance is not gonna increase linearly. So it depends on the like complexity and the purpose of your code. But generally, in general, the more threads you use, the faster your job runs. Okay, so we have the -p. I think it shows the progress bar. So when we do the FASTQ dump, it just shows the progress along the way. And the -big O is the output directory. So remember when we run right here. So when we run the prefetch, we do the -- like output directory. And if you look at the manual of the NCBI, so it also says if you put -big O, it's the same. So -big O is the same as -- output directory, but it really depends on the function. So you need to refer to the manual before you use this sort of shortcut. So we want to put the output to this folder, and this is the input. So the input is within this folder, we have this SRA file, which we just downloaded, and that's the input. So that's why I want to change back to change back to our home directory first, because every time okay, let's try this. So if you don't type the folder name right here, if you look at our directory, our current directory, there's no such a file called this, right? So if you simply hit this, there's no way you can find, so you couldn't access this specific file. So you need to let the function know that this file sits within, oops, I probably need to paste again. Let me copy again.
I think there might be one more directory to go down for the file.
Okay, let's see. Yeah, okay. So for the input right here, so we want to indicate where the SRA file sits. So we are going to this directory, and if you hit double tap twice, oops, okay, so never mind. So if you hit the tab, then it fills the folder for you, which means it has another folder in there, and then we can type in the file we want to download, we want to convert. So this one. So basically means this directory is pretty hard to explain. So this directory is currently one of the directory we can see, and this is a subdirectory within this, and then this SRI sits in the subdirectory right here. Yeah. Okay, so you can run it. So that's the progression bar right here. So I think it's only a couple of minutes, and then we have the FASTQ file sitting in the output directory right here. So when you run functions in Linux, the directory matters, because if you are in this directory and you want to direct to some other file in another directory, then you need to provide a correct path to the file. Otherwise, the Linux won't recognize that file.
For the Windows people, if you go to that folder, the output folder in your MOBA Xterm and you hit refresh, you should be able to see the FASTQ file piling up there while it does the concatenation step, that concat step.
Yeah, so for, oh, by the way, so for Mac people and also for Windows people, so if you want to download or upload the files to the computing server, so there's a way to do the like CP, SCP, as Dr. Fungan introduced last week. And there's another way to do so. So for the mobile external for Windows, it should be easy. You can drag and drop. You can see all the folders in the Linux machine. And for Mac guys, you can use the typical FTP. So I mean, there are multiple FTP software. My personal favorite is the FileZilla, but I know it's probably like 3 to 5 different FTP free FTP softwares you can use. So if you download the FileZilla and just simply type in the IP address you have, I'm putting the SFTP because it's a secure FTP. So most of the machines in our campus are secure FTP. And you put in the login, your account name, your password. It's the same as you log in to the virtual machine by the command line. And then you do the connect. Then you'll show left-hand side is your local machine. So that's everything sits on your computer, your own computer. And right here, that's all the files on the server. So you can click in, so you can see all the folders, all the files in there. You can drag and drop single or multiple files. So you can upload and download files fast with the FTP. So you can do the SCP through command line, or you can do the FTP, the FileZilla. So my personal favorite is the FTP FileZilla because you can see all the sort of hierarchy of your directory, your folders. Yeah. So Mac has an embedded the FTP client. I think it's like a Go server or something. Dr. Chen probably has a recommended the connect to server or go to folder. So it has a embedded FTP, SFTP, the client. And the other one, CyberDoc, is Dr. Chen's favorite. So you can try, any of the FTP client softwares. Okay, so this one's done. Yeah, FileZilla I think works for Windows as well. So you can install the FileZilla or I think pretty much all the FTP software on both Windows and Mac. But mobile external already did. Yeah. Okay. So if you do the FASTQ dump, so it's already finished. So let's take a look at the output. So we can do LL again. Now all the results. So we indicate the result to be saved in this directory. So we can cd to this directory, and then list again. So you see these three are new. So originally we only have this folder containing the SRA file. Now we have the FASTQ file. So you see the 1 here and 2 here. So the 1 is read R1. So it's remember it's like pair end. So each DNA fragment, each cDNA fragment will read from both ends. So that's the first end, and that's the second end. So if you want to take a look at the contents, there are several ways. So you can do less, LESS, and then the file name. So it pretty much apply to most, I would say most of the text files. So you can use less to visualize it. So you see as I introduced, you have the first line is the identifier of the read, and the second line is the read sequence, the sequence read, and the third line always comes with a plus sign, and then the fourth line is the quality, the per base quality associated with the sequencing read. So these 4 lines are the first read, and these 4 lines are the second read, so on and so forth. And you can use I'm not sure whether scrolling works, but you can use your keyboard to go down, go up or go down with the less function. So you can view from the top, and you can go all the way to downward. And if you want to quit, you can put Q there. Good question. So if you want to know like which character respond to which quality score, you can simply Google the quality, I think it's called Phred score, PHRED, the Phred score of sequencing. And then I think there's a lookup table for you. And for most of the quality control software, it generates the stats or even pre-filtering, the trimming based on the quality scores. And for most of the, I think for most of the Illumina sequencing, it's like, we're not doing too long, like it's like 100 bases. So the quality is typically high. I mean, the entire sequence, the quality should be satisfactory. Yeah, but there are software to filter. See, I don't think so. So the ring score should be the quality of your RNA sample. So you have one score per sample. Please correct if I'm wrong. And for the sequence read, we have a score associated with each base, each read. For each read, for each base, we have a quality score. So the score basically the quality of the fluorescence signal when we detect because Illumina detects per base based on the fluorescence signal. So I think that's the quality of the signal, the fluorescence signal associated with that particular base. Okay, so so we just did the less on this file, so you can look at the file. So if you want to query, just put Q, just single Q, and then you go back to the original directory. And another two ways I think Dr. Feng introduced last week. So you can also do head, and you can do dash like 20 lines and of this file. So you can view the top 20 lines of this file. And you can also do tail, 20 lines, and it shows the bottom 20 lines of this file. And if you want to know like how many lines are there in a specific a document or specific file, you can do the WC-L. WC means word count, I think. -L means lines. So you can calculate. I'm not sure how long it takes on our machine. Let me try first. You guys can hold a bit. So it tells you how many lines we have. Oh, that's fast. That's fast. Okay, you guys can try. So the WC-L and this file. So it tells you it's let's see how many. It's like 94 median lines, but you need to divide it by 4. So it's roughly like 20 to 25 median reads in this sample, in this specific sample. So WC-L give you the number of lines content in this FASTQ file. Okay, so now we have both the R1 and R2, the pair and the FASTQ file, and then we can go back one directory, go back to the home directory. So it's right here. So you can cd ... means you go back to the directory one directory, or you can cd tilde, either way. So just go back to the home directory. And next we'll be running the Salmon. So let me briefly introduce this function first. So Salmon is the program we are using. And that's a good question. So when I downloaded it, I didn't find a kind of introduction to that. And that file is typically small, but for this file it's large. I'm not sure. I thought it's the like filtered FASTQ or something. Henry, you have any idea on this?
So this is honestly a little bit of a guess for me, because usually I don't do anything with that file. But when you split your data into your mate pairs, so your _1 and your _2, there are often some reads that don't have a mate, and those will go into a separate file. So they're called singletons or single read or single reads. So I'm pretty sure the default for faster QDump is to just write those into a separate file. I'm not sure why it's so large for this one though.
Yeah, for typical, yeah. So the other one, the other sample, I was trying the other day. So I think it's only like I would say like 5% in the size of the other. I mean the _1, _2. So this one's supposed to be very, very small. So that's why I guess it's something, as Henry said. So it's probably something not important, pre-filtered. And this one, I'm not sure why it's so big. Okay. So here's the Salmon function we'll be using. So function is Salmon is the function we're running today, and the mode the parameter is the quant. Quant means it's quantification mode. And -L is the library type. I have a slide. If we have time, we can probably cover it later today. So -L is the library type. So we typically do A means automatic library. So if you have a very specific library setting, you can do the typing specific parameter here. But most of the case, you just type in the A for automatic, just a default setting. And -1 is the first fast queue you have. So that's the first fast queue of the pair end. -2 is the other, I mean, the paired fast queue for the same sample. Okay, so these two samples just generated from the fast queue dump. And we have the -- validate mappings as a default setting of Salmon. And the -I contains the index. So the index is what I mentioned. So they look for the specific sequences within each transcript. So the -I is actually a database provided by Salmon and by other third-party users. So the -I are pre-computed. So it's a database in our, I mean, in this function. So I think we already have the this index downloaded in your virtual machine. And -P means how many threads we want to use for Salmon. So you can see even for the same, I mean, threads, you can have -P right for Salmon. For FastQ dump is -E. So it really depends on the parameter. I mean, the naming the programmers defined in the original function. So you need to always refer to the document to see what are the parameters and what would be the potential settings associated with the function. And then it's a small o, little o is the output. So it's not a big O here. And we want to put output in the test Salmon out and this folder. So let me copy this. Okay, I'm pasting to the chat box. Okay, so I think it's good. Let's see. Yeah, so just paste the code to your virtual machine and then hit enter. So it runs within like 2-3 minutes. It's really, really fast. So for typical like top hat, the alignment based methods, it's a couple of hours for each sample. So it's really, really fast for Salmon to do quantification. Okay.
It looks like you got stuck reading from disk. This is so if anyone's noticing the same thing happening, it should be a little bit faster. Okay, it looks like it got through. Some people might find that it got really slow, and you just have to wait a couple minutes. It's an issue with Azure that they're having today if it ends up being very slow.
Yeah. Okay, so I can, while we are waiting, I can go to the output of the Salmon, just save some time. Okay, so the output of Salmon is really actually very, very simple. So it has a bunch of files we'll be seeing in a couple of minutes. So it has like all the parameters and log files. Those are not important. So the main output is the quant.sf. I Googled it, but I didn't see like what SF stands for, like Salmon file, maybe something like that. And it's the quantity. It's the abundance of genes or transcript, depends on the database we're feeding. So the index we're using are for the transcripts. So it provides the quantity, the abundance of each transcript. So here's a snapshot. So it's a table. So you have the ENST are the transcripts. So you have different transcripts right here. And you have the length. The second column is the length. So the length of the transcript. And the third column is the effective length. It's quite unique to Salmon. So it says the length of the transcript adjusted or normalized with the GC content, also the non-specific sequences right here. So it basically adjusted or sort of normalized length. I tried to take a look, and most of the transcripts, the effective lengths are very, very close or equal to the actual length of the transcripts. So that's the length. And the fourth and the fifth columns are the most important, the results. So the fourth column is the TPM. So I guess most of you heard the TPN. So TPN is a way to express the expression level of each gene or each transcript. So it's the full name is a transcript per million. So it basically normalizes the read counts based on the total number of reads and also the length of the transcripts. So we can take a look at the results later, but if you add up all the transcripts in one sample, the sum will be 1 million exactly. So that's how the TPN does the normalization, and the number of reads right here will be the reads we want to use for DSeq. So DSeq takes the number of reads as input, and if you are doing you're using the data for other purpose, you want to do draw the expression level. You want to draw some people do the scatter plot for the expression level. You want to see whether the gene is expressing or not. Then you can probably use the TPM because it's more normalized the measure. But if you want to do the differential expression, I think most software takes the read counts as input. Okay, so now the Salmon is done from my end. I'm not sure how you guys are doing. So let's see.
Chris, would you mind just really briefly showing the mapping rate part? So that's right above that last line that you have displayed right there.
You mean the file or the message?
So it's the one where it says if you go right above where it says finished quantifying library, yeah. So right there it says mapping rate.
Yeah.
I just wanted to point this out really briefly. So this is something that is actually a really important quality measure for a lot of RNA-Seq, which is just what percentage of your reads actually were able to map to the transcriptome, to the human transcriptome or the human genome. Just because it can be an issue if you have contamination in your sample, or for example, if you have another, like a mouse or a fly sample mixed in, that will very strongly affect that mapping rate.
Yeah. Okay. So now all our results are in the test salmon.out. So you can see the test. So in this case, if I hit tab, you see it stops right here because we have two of the files with test underscore capital S. Then you need to indicate which folder you are going to. So I just type in A, then you do tab again, you'll fill the entire directory name for you. So if you CD there and do the LL, then you see the one sample right here. So you can do CD again. So you see a bunch of the information and the logs associated with the run, and the most importantly is the quant.sf. So you can view it by the less quant.sf. Then you can see the transcript, pretty similar to what I just showed. The transcript, length, and the TP and the read counts. Okay, so you see some transcripts are expressing. Okay, so that's it. And we already have from the box folder. So you guys can keep this running if it's not done yet. But from the box folder, I think you already have the six samples done. I mean the Salmon outputs of 6 samples. So from the box folder, I can probably show you right here. So from the Salmon out folder, so Henry already did Henry already did 6 of the samples.
It's
okay,
it's in the chat.
Okay, I already downloaded. So you can take a look at the chat. There's a link. So I already downloaded everything. Let's see. Okay. So week 11. So if you download it, there'll be multiple files. So I'll walk you through the R code to generate assignment as. So we'll have a short code to sort of load and merge all samples together, because as you see, each of the sample, if you look at the Salmon out, so there are 6 samples we already ran, and each of the sample you have an individual folder and contain a individual quant.sf file. So each quant.sf file only contain the expression level of that specific sample. So we need a short R code to load all the quant.sf files in, and also merge everything together into a data frame or a table for DSeq. Okay. So everyone can download the week 11 folder, and you have the Salmon out and also this code, the import and analyze.r.
Yeah, I updated the data probably 30 or 40 minutes ago. So that's actually that's the older version. So if you could all download the latest version, that would be really great. You just download the entire week 11 folder, unzip it, and then open the R project file. That should bring you right to where you need to be.
Yeah. So we're still having 6 samples, right?
No, it's more now.
Okay. So yeah. So I can quickly explain this code. So this code is written by Henry, so courtesy of Henry. So I can quickly walk you guys through this code.
You shouldn't need to do the set working directory. If you just open the R project file, Chris, though, could you download the latest one?
Oh, okay, yeah. Let's see. Yeah.
Awesome.
Okay, so this one. So it's in the import and analyze.r. Okay. So for first, I think everyone are with me now. Is with me now. So for the first time use, if you're using so the package we're using today is the TX import. So I think TX stands for the transcripts and genes. So it's to import the Salmon and many other files together and really prepare the transcript or merge the information of transcript and genes. Okay, so it's a bioconductor package. So for first time use, you need to install the TX import. And after you, let me run line by line. So after you already install it, then we can library all the packages we need. Okay, so we're also librarying the ensemble database for Homo sapiens. So it contains the gene annotation files like transcript ID, gene ID, pretty much all the information associated with the genes, and it's version 86.
Sorry, Chris, would you mind actually zooming in a couple?
Yeah. How should I do that?
I honestly don't know how to do it on Mac. Does anybody who uses a Mac know how to zoom in?
It's Command plus.
Okay.
Okay.
Thanks.
Doesn't work.
Works.
Oh, it works. Oh, okay. Okay, that's good. That's good.
Oh, the Mac has all the trick. Henry, sorry.
No, that's cool.
Okay. So after you library the Homo sapien database, you can just do the columns. So you see all the information are the information contained in this database. So we'll be using the ENST ID because as you see the Salmon output per row, each row is a transcript. So it's an ENST ID. So we'll be using the ENST and also the gene symbols. I think somewhere the gene symbols in the ensemble database. Okay. So this line. we'll extract we can visualize. So this line will extract the gene ID, the transcript ID, and also the gene symbol. Transcript ID is the same as the output of the salmon and the symbols. So I think you have a line, yeah, to remove the gene ID right here, because we won't be using the ENSG IDs today, so you can remove it. Okay, so then this couple of lines reads or to find all the samples that are in the salmon output. So if you run the first line, okay, I can probably explain this. So this is the list directory. So it lists everything within the salmon.out. So remember salmon.out we have right here. So we have eight folders right here. So it lists the names of those 8 folders. So let me run this, then you'll see.
And just to be clear, while he's running that, so what he showed you on the server, we did that for all of these samples individually. So if you wanted to do all of these yourself, you'd have to go back and run all of that command he showed you on the server for each of these samples.
Right.
And then download it.
And for each of, yeah, then download it. Yeah. Okay, so if you run this line, you see the samples. So these are pretty much the directory folder names, the directory names within the salmon.out. So for each salmon run, we have a new folder. So for each sample, we have a new folder. So it basically lists all the folder names within the salmon.out. Basically all the samples we have. And then the next line. Okay, so next line, want to prepare the path. So the path we have is actually the salmon out and all the samples, because we have the sample name and the quant SF. So you can see now we have I probably couldn't. Yeah, right here. So you see, we have all the directories, all the paths to the 8 samples, 8 quant SF samples we want to use. And then we add the names to this, so you can see. So we have all the paths and also the names. Names are critical for the TX import. So the names will be used for the column names after we merge the everything using text import. Okay. So this line, so it just to check whether the file exists. So should exist. So all the A files are there. So it could be read by R. Okay. Okay. So then we have just to set up the metadata frame. So that's for the TX import. So now we have the A samples. I think it's already ordered in the sense that it's untreated and treated sense. So we repeat it for 4 times. So it's untreated, treated, untreated, treated for 4 times. Then we have A samples. Then we run this. Okay. So the key part, the TX import is actually here. So we spent some time to prepare the inputs to the TX import. So the TX import actually runs we need to give the files the path to TX import, and the type is the salmon input. And we have the TX to gene variable. So that stores the ENST to gene symbol table, and also the ignore TX version. So ignore TX version is something I can probably go back to the terminal to show you. So in the ENST, so in a quant.sf file we just had, so it has everything with the dot something. So that's the ENST. This part is the ENST ID, and this part is I think it's the version or something. So for the same ENST, you can come up with different versions. So that line, that parameter in R, that parameter removes the, oops. Okay. So where it is, yeah, this parameter, it removes the version. So it ignores the dot something version. So when you look at the ENST ID, so then we can run this. Okay, somehow I probably went to the wrong RStudio. Oh yeah, I went to a wrong RStudio. We have A samples. Yeah. It's not all zero. So if you, I probably have an open version in Excel somewhere. Yeah, I have. Okay, so if you open the file in Excel or in R, so you see I mean this figure is the relationship between the length and effective length. That doesn't matter too much. But if you look at I can show the TPN. We'll see. So TPN typically range from 0 means it's not expressing, to 10 or 20. So you see like rare genes that are like over 100 or thousands, but for most of them, I think 90% of them are below 20, 30. So you see those are expressing genes, and a majority of them, probably more than half of them are not expressing. So they'll be zeros. Okay. Yeah, so then after you load it, then we have the TXI. So then we can do the DE-seq. We can prepare the DE-seq data set from using the DE-seq from TX import directly. So you can just run this line. Then we have the DDS. We have the DE-seq data set for you. So from this on, you can use the typical DE-seq, the differential expression analysis, which was covered in previous weeks, and also the PCA. I probably won't be running this part because we probably don't have enough time. So this part are it's quite fast actually. So it shows the PCA of the sample. So we have four of the untreated cells and 4 of the treated cells in the PCA plot. Yeah. Oh, Henry actually provided the gene analysis. So you have pretty much everything needed here. I thought it's the part of the homework.
No, I figured that this part's only a couple lines, so I might as well show this part, but yeah.
Yeah. Right, so that's pretty much the article needed to import and to merge all the single sample files from Salmon, and to prepare the DE-seq variable DE-seq data set needed for the subsequent differential expression analysis. Okay, and there's another shell code right here. So in the box folder you download, there's a shell code, so you can build it by anything. I mean, in Mac it will be text, and everything related to like text edit or the BB edit, everything a text editor. For Windows, I'm not sure. You have a notepad or something, right?
You can actually open that in RStudio.
Really?
Yeah, it does the color and everything. Yeah, it's pretty nice.
Okay, I can drag and drop. Let's see. It probably doesn't recognize it. Let me.
Yeah, you can do the Control O from, or I guess Command O from RStudio to open it. So like if you actually look at the file panel in RStudio, you'll see it there. So yeah, so the files panel, it'll be in there on the bottom right hand side.
Yeah. So it's pretty much the same codes we use to run individual samples, but here's just a way to just show you how if you have multiple samples you want to run, so you can list all the samples right here. So I think all the A samples Henry just did. So here's the samples, and here's a for loop for Linux. So every time it runs the A sample right here, so you extract say for loop 1, so you just extract the first sample as a sample parameter, and then everything here runs the exact same codes as we just did for individual samples. So this for loop just do all the samples automatically for you. So after we run it, so everything, all the files, all the outputs will be in the Salmon output and under the name of specific samples. Yeah. Okay. So I have a couple of slides to conclude on this part. We have 10 minutes. Henry, do you have anything to add?
I was going to add that in the R script it says that we were going to do hands-on activity, but I don't think we'll have time for that, which is okay. Basically, for your final project, which we'll talk about next time, we're going to basically be doing this, but you're going to find your own study to do this with. So the hands-on activity is going to give a little bit of practice with that, but you'll get practice with that when you're doing the final project anyways.
Okay, so I have a couple of slides to conclude this part. So for Salmon, we pretty much use the default setting. So there are more, if you refer to the Salmon manual, the web page, there are probably like 30, 40 parameters for you to tune. If we don't specify any specific parameter, then we use the default setting. So there are many of the settings you can set, especially for the alignment. I mean, other alignment tools, you can set the alignment criteria, the stringency of the tools. So there are a lot of parameters you can check. So for Salmon, we're using the -L. It's a library type. We use the automatic, I think. So it's A here. So it's actually have other parameters to be set. So you have the inward, outward. So I'm not saying too much about it, but so it basically means when you do the pair and whether you are reading from the ends towards the center or from the center towards the end. I'm not sure why we have the outward sequencing. Dr. Chiu can probably comment. So I'm not sure why we have the outward. I thought that all the sequences are the inward. So it reads from both ends and all the way to the center. So
just different protocol. So very rarely we do the outward, but someone want to do that.
Yeah, so if you have an outward sequencing, you can specify this in the -L, the library type. And also, I think most of the RNA sequencing now is the stranded. So you can specify this here. So the difference between stranded and unstranded is we have DNA is double stranded. So, but messenger RNA mostly is single stranded. So when we do the convert it to the cDNA, we convert it to, we generate the complement strand. So it's two strands, double strand. So if you do the unstranded sequencing, you sequence both the messenger species you have and also the complement species generated from the reverse transcription. And by stranded sequencing, we will only sequence that expressing strand, the messenger strand, instead of the other complement strand. So I think there's a trick in the reverse transcription. You can add something in the reverse transcription to label the messenger RNA strand you want to sequence. So that's the stranded part. And you have other, like to specify whether it's the forward or reverse strands, more specific parameters you can do if you have a specific design in your sequencing run. And it also has the way to run the single end reads, because the examples we did today was the pair end. So for each of the sample, we have R1 and R2. So if you only have a single end, which means you only have like single FASTQ per sample, so you can do the -R right here. So it specifies you are running the single end mode. So you just only need to feed the function a single FASTQ file. So it's not R1, R2. In our example, we have -1, the FASTQ R1, and -2 FASTQ R2. And if you have a technical repeats, you can do the -1 in pair end sense. So you have a 1, 1, 1, 2. You can put all the technical repeats. So say if you have 2 repeats for the same sample, so you can do 2 repeats right here, 2 repeats right here. So I think it summarizes everything into one single output. So it basically summarizes the repeats for you. Okay. Yeah. So that pretty much are the alignment free, the pseudo alignment pipeline represented by Salmon covered today. So there are, as I mentioned, there are another like workflow of RNA sequencing alignment, as Simon asked. So that's alignment based. So I look for some of the papers to compare the consistency between different alignment tools. And those consistency were compared only on the gene expression levels, because for other like mutation calling, like fusions and many other applications, the alignment free Salmon couldn't do it. So this comparison mainly based on the expression levels obtained from different types of RNA alignment tools. So I Googled several papers. They benchmark these tools in many different cell lines, many different species and different conditions. But most of the conclusion is that you see the scalar plot of gene expression obtained by different alignment tools. The consistency was pretty high, so mostly above 90.98 or 0.99. So in this paper. And also this paper compared the different alignment tools. Here's the Salmon, here's the top hat cuff link. So to the qPCR, which is the ground truth. So you see pretty much the correlation is high. It's R squared, so in a square sense. So I think the original R is 0.98 or 0.97 somewhere. And the only difference is probably right here. So for those lowly expressed genes, so I mean, in the qPCR sense, for those lowly expressed genes, if you use alignment tools or sequencing, it's probably much higher. The sensitivity is higher for those lowly expressed genes compared to qPCR. That's the only difference I observed. But the dramatic difference is actually in the computing time. So as we just did, so for Salmon, each sample takes only like I would say like much less than 5 minutes. But for typical top hat and other tools, it could take multiple hours. I think the unit here is hours. So you can see it's dramatically different for the alignment-based methods compared to alignment-free methods in terms of the computation efficiency. Okay, so there are many alignment tools out there, and you probably want to ask which one is the best one. Okay, so I'll conclude. So I tried to Google some of the large projects, like what alignment tools they use, and also our sequence. You could do the sequence and bioinformatics analysis in our course. So what would be the alignment tools? So in our course, as far as I know, we use the top hat tool and HiSet and also RSEM. Those are alignment-based methods. So after alignment, you'll get the chromosome and location to indicate where each of your read come from. So that's our the course. And I think for Dr. Zheng, his lab used a lot of STAR to customize STAR to identify the gene fusions. And for TCGA, I think they use the STAR as a standard for their RNA-Seq analysis. And for Recon2, Henry just mentioned a couple of weeks ago, they use a rail RNA. I'm not familiar with this one, so they use another alignment tool. And for ARCH4, so they have reprocessed almost, I think, 500,000 samples of RNA sequencing. So they use the Callisto. So Callisto is kind of similar to Salmon. It's an alignment-free tool. So it's quite fast. So it's probably why they adopt this method for their project, because they are dealing with a huge number of samples. So everyone has their own favorite aligner, and different aligner is probably optimized for different tests. So it really depends on whether you are doing mutation calling or you simply want to quantify gene expression. So different aligner can give you can have different strengths in different tests. And the key notes you need to keep is that for each of the project, each of paper you're writing or you're preparing data for, make sure you use the standardized aligner. Use single aligner and consistent parameters. So don't mix and match the aligners or the results from different aligners or results from different parameters, because that will create sort of batch effect from the computational side and also the concerns from the reviewers. So make sure within the same project use the single aligner and as consistent parameters as possible. Okay. Yeah, so that's the last slide I have. So everything beyond the gene expression quantification, if you have long read RNA-Seq, so it's quite hot recently. So for long read RNA sequencing, they have their own alignment tools and also mostly to study like fusions, transcript, splicing events, all those. So there are another branch of alignment or analysis pipelines for those long read RNA sequencing. If you're interested in splicing and fusions, those couldn't be done by Salmon, so you need to look for other standard alignment-based methods. And if you are dealing with whole genome sequencing or whole exome sequencing on the DNA level, so I mean, those are very, very different applications on the DNA level compared to RNA. So for those, pretty much all of these other applications, you need to look for other RNA-seq pipelines than the Salmon. Okay, that's pretty much it. Any questions? Yeah, I think Henry and Dr. Chen pretty much answered all the questions.
I don't know, I guess Chris kind of touched a little bit. So we also talk about, so it's really worth watching what would be the new alignment program for the long read thing. Oops, sorry. So in that case, those probably, if anyone want to work in those areas, if computational kind of oriented, that could be one of the direction that you may want to pay little attention. Yeah. So I don't think Chris, you cover any this part. Yeah.
Right. So for long read alignment, long read will typically do alignment. So for long read alignment, the strategy is probably very different from the short read, because short read is like it's only 100 bases, so we can require like a 99 or 100% or 100 of them to be perfectly matching the reference genome. But for long reads, it's more noisy, and it could contain much more like the splicing and fusion, all the events. So there are some aligners specific design for long read RNA sequencing. So I mean, they're using quite different strategy, and still I think much more methods are gonna be developed very soon. Right. And all the applications of the long read sequence, like splicing, fusions, everything, all those tools, we are currently kind of having a project working on long read. So I think the analysis pipeline is not that mature yet compared to the short read Illumina based sequencing.
Were there any more questions?
Yeah, I had a question still about the output from Salmon. So I'm just looking at the quant.sf file that gets spit out by it. Could you explain what TPMs are again? I don't think I understood that. Like what that is and what the difference is to like number of reads.
Yeah, good question. So the number of reads is simply the number of reads aligned to the transcript. So it's supposed to be integers, I would say. But for because we are counting the number of reads aligned to the transcript. But if you look at the Salmon output really carefully, the number of reads are not exactly integers. So sometimes they have a floating point. So I think they have a statistical inference or something to correct the number of reads. But suppose the number of reads need to be integers for most of the alignment tools. And the TPM is a sort of normalization. So it normalizes two things. So the first thing is the length of the transcripts. So the longer a transcript is, so if you have a very long transcript compared to a very short transcript, we are randomly sampling the RNA species from the total RNA, right? So if you have very long the transcripts and the number of reads you align to the longer transcript is higher, so you will have much more reads aligned to the long transcript compared to the short transcripts. So that's why we need to adjust the effect of the transcript length. That's first. And we also need to adjust the total number of sequencing reads per sample. Say if you have a sample you sequence by 1 million reads, and you have another sample, the same sample is sequenced by 2 million reads, then the gene expression will be doubled for those 2 million reads. So that's another factor we need to adjust. So the TPM basically adjusts for the length, and also I think it divide each of the read counts by the length, and then sum all the values together and then divide it by million. So that's why it adds up to the million. So if you open a single quant.sf folder and you look at the TPM column, you can click on the TPM column and sum the entire column across all transcripts, it sums up to be 1 million exactly. So
Okay.
that's a normalized, I would say normalized measure of gene expression level compared to read count. Read count is a very raw counts of reads per transcript.
Okay, and which of those two gets put into DSeq?
DSeq, I think you use the read count.
Okay.
Yeah, I didn't hear. It's now using the read count, using the transcript.
Read count per transcript.
No, the TPM stands for transcript per million transcripts.
Yeah.
It's not per million reads, so that the unit is actually transcript. So you have to first assemble the transcript, then count total number of transcript, then divide each particular transcript by the total number of transcript, and multiply million afterwards. So that's kind of the way of doing it.
Right, but the matrix that gets fed into DSeq in R, is that the matrix of TPMs? Sorry. Oh, that's read counts?
That's a read count. That TPM and RPKM is only used by few algorithms. Most of algorithms start with a raw read count. And a lot of algorithms actually building with, for example, DSeq, you can easily export FPKM in this case. They kind of do the other way around. You can get the FPKM you want to. I don't know DSeq has a TPM. I don't believe though. But you can get FPKM. Yeah. So
right. And I'm not speaking of that. I'm not sure whether you need to run the results of Selman for DSeq. Henry can probably comment on that. So some of the quant.sf result, you see the floating point, not integers in the read counts. So I'm not sure whether it generates problem for DSeq.
Oh, sorry, can you say that again?
Yeah, so for the number of reads, I mean the Selman output, the quant.sf, we have some of the floating points at the number of
right. Yeah, so it's got decimals, right? So DESeq automatically deals with that when you use the DESeq from TX import function. Okay. And it actually also takes the transcript length to like it does this thing where it does a little bit of a correction for the library size based off the TX import. So it's actually they work together. The authors of DESeq work together with the authors of TX import. Some of them are actually the same people. So they figured out a way to optimize that for DESeq, which is why I like to recommend it.
Yeah, but you can convert from all these things if you want. It's just not ideal. So I mean, ideally, if it's a DESeq, I'd probably still recommend just go with the read count and just run to the integer and it goes. But people kind of having all these tools, and I'm sure you can find the tool converting it back and forth all the place. That this is a kind of a very common units that people use. There is actually pretty interesting. If you want to follow the internet thread, then there is a lot of controversy between read counts, FPKM, and TPM, or whatever CPM, and all these things. So but it's kind of fun read if you want to.
Controversy is one way to put it. It's a lot of Simon Anders and Michael Love telling people that they're wrong. It's a lot of people being a little bit snotty on the internet.
That's just the internet.
Yeah. Scientifically speaking, that there's also political correctness involved in there, but it's more in the scientific term. You learn something there. So I'm not saying who is right, who's wrong, but it's kind of interesting.
It is nice because they will give very full explanations. So you learn a ton from reading the posts, but definitely it can get a little bit heated. So, all right. Were there any other questions before we break?
Hey, I just one more. Sorry about that. Again, looking at the quant.sf, I'm seeing quite a few rows where the TPM is non-zero, but the num reads is 0. Is that just because the number of reads is so small that it's past the decimal points it displays, or?
Oh, that's a good question. So you mean the number of reads are 0, but
Yeah.
the TPM aren't?
It's up to like a couple of hundred. Yeah.
It's probably for very short transcripts, maybe.
Okay.
So,
Yeah, that goes back to the effective size. So I mean, you should read the post. This whole story comes back. So the effective length is not length. So you may end up with negative value. So if you do the right calculation. So I don't know how they calculate it. I mean, I know how they calculate, but when you hit the 0, that become a real implementation issue, because if a gene length is less than fragment length, because the effective length is the gene length minus the fragment length. I mean, average fragment length. Average fragment length typically is about 200 base, 260 or whatever. So if a gene length is smaller, then the effective length become some sort of a negative value. So different people have a different way of implementing, and given the implementation, then they will have an estimate of various values. So that's just I think when you goes to the recount 0, just go back and reset the TPM to 0 sometimes. So just this crazy thing, because you somehow divide by the relength in some way, someplace, and then it becomes 0. Somehow they're all 0. You have to give a value in some way. So this is, but it's an argument about what is the effective gene length as well. So everything is, everything has a sort of a scientific argument down back there. Then people giving you some what you mean unbiased estimate about gene length, all these kind of statistical or mathematical definition about what are those values really is. So if you look at the column, I think this has the 2 column, right, called the length and the effective length.
Yeah.
In there. Yeah, so that's what how you get the effective length out of it. Yeah.
Yeah, you have the length and effective. I actually draw the scalar plot there.
Should
I mean,
be identical.
Should be, I think, but it's just slightly different, slightly smaller. The effect
because it supposedly subtract the fragment length.
Mm-hmm. They also adjust for the GC content and some other factors. Yeah,
Right.
but it's slightly smaller than the length, and it's almost I mean, consistent.
Right. And the fragment the fragment length is the estimate from your FASTQ data, from your pair N data. So each run is slightly different in that case. I'm giving you the on average value, but in reality they have to get estimate from the FASTQ file. So it caused some, I don't particularly like, so that's the other thing you may not like TPM, RPKM whatsoever. So that could be the other reason. So because if you could just simply give me the RPKM or TPM without telling me the fragment length, I really cannot get back you the raw recount anymore, because that's become a guessing game. What's the value that you subtract, or have you actually implement the effective length function or not? And it's become a guessing game. So this is actually kind of interesting to some extent, that if you care about the exactly value here. So, but you probably point out some kind of a unique thing there. It is tricky though. To seriously understand how they do it, I guess you have to open up their code, look at it. Yeah. So I don't know how do they do it. But everything is very well defined. You have it's understandable.
So for the sake of time, because we're now quite a bit over time, I think that maybe a couple people had questions, but let's go ahead and just wrap up now, and then we'll hang out for a few minutes for people who wanted to ask any more questions. So now you've got an idea of how to use a Linux server to get the raw data and to process it into the read counts that you can put into DE-Seq to do your analysis. So you've got the whole thing now. You can start from raw data and go all the way to the point where you've got your biological interpretation. So next time, Dr. Zheng is going to talk about his research and what you can do beyond this simple sort of gene expression. But then we're going to start the final project, in which what you're going to be doing is you're going to be finding a study. You're going to be doing this analysis all the way from the raw data down to your biological interpretation, and then you're going to present that. And that's basically going to be a wrap for the workshop. So thank you all for sticking with this. And yeah, we'll see you next time. Again, stick around if you wanted to ask any questions. But for those of you who want to head out, we'll see you next week.
Yeah, bye to everyone. I have to go to another meeting, so just.
Oh no, it's almost 7.
To leave. You can ask Chris and Henry more questions.
All right. So I mostly because I saw Aola and Muku, you turned your cameras on, so I didn't know if one of you wanted to ask a question or anything like that.
I had a quick question.
Okay.
So I wanted to clarify, because I don't know if you guys mentioned this, and I don't know if I'm missing something about the concept of Linux, but so I thought Biniconda, is that a Python installer? So are you using Python in Linux? Is that how we're downloading these packages through Python and then using it in a Linux shell, or how's that working?
So I can actually show you this on Windows. Basically, what you do, and I'm going to show this here. So this is my MOBA Xterm. This is me connected to the lab server, but that's going to quit in just like a second because I told them to shut it down. So I have Anaconda installed on my computer actually, and it should just take 1 second to show up here. Yeah. It always takes a second to load. But basically, Anaconda is a package management system. So it is based on Python, but it does a lot of other stuff. So it's language agnostic for the most part. What it's going to do is it's a way to organize all of the software in your computer into environments. So right now I'm in a base environment here. And so if I wanted to create a new environment, I'll just do this conda create, and then I'll give the name of the new environment is my environment. And then I can create this new environment and load into it whatever software I want. So it's asking me, do I want to create this? Sure. So I created it, and then now I'm going to activate it. My env. So now you can see it switched from base to my env. So I think I still have, no, I don't have Prefetch anymore because now I'm in my new environment that doesn't have this software, but I can install it. So if I do conda install C bioconda, because I think that's where Prefetch is from this channel called bioconda. So you specify the channel where you want to get it from. It probably isn't that one. All right, so I'll show you how I actually do this. I'll Google it, conda install Prefetch. Yeah. This is how I actually do it. So I come over here to the first thing, which is, so when you Google this, the first thing is this SRA tools. And then I'll just copy and paste this string here. So yeah, I guess that was the right one. So I'll copy, I'll paste that, and then I'll install it. And that was actually the entire, and then hit yes. That's the entire installation process for Prefetch. And I guess it takes a second to unpack all of this, but basically it's going to install that. And if it depended on anything, if there was any dependencies for this software, it would have also installed that. Now I'm on Linux here, but you can do this just as easily on Mac and on PC. That's the beauty of Conda, is it's actually language agnostic. So I see now I've got Prefetch is working. So this is what Anaconda looks like on Windows. So you can see it here. And you can actually open this Anaconda prompt. And so when I do that, it's because it's Windows, it's in PowerShell or whatever language Windows uses. But basically I can do the exact same thing. I can do conda create my env, and I can install Prefetch here as well and use it the exact same way. The only difference is that some of your shell commands like ls, I don't think work. You have to know what the Windows version of that is, but it's really okay. Like, as long as you've got a cheat sheet, then you'll be able to use this in the exact same way. So you can just see here, it works the same.
So you just used it on your MobileX term. Is there any way, did you have to do anything to like link your local computer's Anaconda install to your MobileX term session? Like, how do those communicate?
Oh, this isn't on MobileX term. This is just on my.
No, but just now before you went back to Windows.
Oh, here?
Yeah.
Oh no, I mean I'm connected to a remote computer. So
that
I had
computer
to.
has Conda?
Yeah, so you can see here, this is actually the installer.
Ah.
So to run this installer, you just hit bash. So bash is the command that you use to run shell scripts. So this is a shell script that says .sh. That makes it a shell script.
Yeah.
So you do bash, the name of the script, and then you run it, and it gives you this like installing prompt. It gives you like the license agreement and stuff, and then you can just scroll on through that. You don't have to read that. And then it says, please answer yes or no. Do you accept the licensing terms? You hit yes, and then it'll install everything for you. Okay, it says it's an error because I already had it installed. And
you did that on our virtual machines before we showed up, or are those like pre-installed?
Right. Yeah. So that's what I did. I did this on your virtual machines, and then they built so they made an image based on the machine I had, and then they replicated it like 25 times so that you would all have the same thing as I had.
I see. Okay. Cool.
Yeah, but that's how you install Miniconda. And actually, if you look online, if you go here and you say, install Miniconda 3, if you Google that, the very first thing that comes up is the page where they show you basically the installers. So the Linux installer is this one. So I just downloaded this file to the Linux computer, copy that address. wget is the way that you download things from the internet, or at least the way I do it. wget, and then now I've got this file is on my computer, except it said .1 because I already had one of these, so it renamed it .1, but you get the idea basically. So that's how you would do it from scratch on a Linux machine. On a Windows machine, it's a little bit more, I guess, a little bit more user friendly because you can just use the installer that you download from Chrome or wherever you want to download it from.
Okay, yeah, that makes a lot more sense because I'd used Anaconda before, but just for Python, and I didn't know that it could do that. So
it's pretty nice.
It does R too, right?
Yeah, it does. It's language agnostic. It does every language. There's very few things that you can't do with Conda. So I use it for everything personally. Okay. Did anybody else have any like questions or things they wanted to, I guess? Yeah.
I just have one real quick general interest one. So sure. What we did on MobileX term today, what kind of computing power was involved there? So if you did this on like a, fairly strong home desktop computer, are we talking this would have taken us an hour or like a week, or like what's the computational expense here?
I actually ended up regretting doing the whole virtual machine thing because I realized that your personal computers, it wouldn't have been nearly this fast because we had 16 cores here. Most personal computers only have 2 or 4, but it still would have been pretty fast. It would have been like 20-25 minutes to do a single sample. So it's totally doable on your laptop. The issue is more the RAM, so the amount of RAM you have. So we had 64 gigabytes of RAM on these virtual machines. Your computer probably only has 16 or 32 at most. So that's one place where you can run into issues. Now, Salmon is smart enough to account for that, but part of the way it accounts for that does slow down the process. The other thing is storage. So just doing the shell script to download those 8 files and run them, it only took 30 minutes to finish running that entire shell script where it downloaded all 8 of those and ran the whole pipeline on them. But it did eat up like 200 gigabytes of storage. So you've got to have space on your computer, or you can just be smart about it, which I wasn't, but you can be smart about it. And then in your shell script where you're automating your pipeline of downloading the files, running Salmon, and then getting the output, you could also include a line that deletes the raw files after they're finished. So you download them, run Salmon, and then once Salmon's done, delete the original FASTQ and SRA files, and then you won't take up so much storage. So that's one way.
So the output files are fairly small, I'm assuming. It's the raw files that are giant.
Right. The output files aren't more than 10 megabytes. So yeah, it's the raw files that are massive.
Okay. I mean, yeah, because like a $500 home desktop CPU will have 16 cores now. Like a Ryzen 9, yeah, Ryzen 9 3950X has 16 cores and 32 threads.
Okay, I mean, yeah, if you're getting like a Ryzen Threadripper or whatever, yeah, sure.
No, man, Threadripper is like more than that, but just the base Ryzen, the like home desktop, not high-end desktop computers, they'll have the highest end one has 16 cores and 32 threads.
Wow.
Yeah, and they all run at like 4.7 gigahertz. It's insane.
What is the average consumer doing with that kind of power?
Video encoding.
Okay, that makes sense. Yeah.
Content creators use those. Yeah.
I would not recommend. So if you're considering doing any kind of work like that, I would recommend talking to Dr. Chen about getting access to our server, the server that we have at the university, because it's made available for people who are doing this kind of research to be able to use it. So I think Aola I think you just got access like a week or so ago. That one's got 192 cores and 500 gigabytes of RAM. It's pretty good. You're sharing it with other people, but for the most part, you're not going to run into too many issues there.
Unless you're Adrian.
Unless you're Adrian. That was very funny. Yeah, I think everyone's realized, and I think Muku, I think you had this experience as well, and I also had this experience when I first started. You're going to make a mistake at some point, and you're going to use up all the RAM, or you're going to use up all the cores, or you're going to use up all the storage. It's just kind of inevitable. And then you'll get an angry email from Dr. Chen or from me or from whoever's on the server at the time. It's kind of how it works, unfortunately.
Yeah, that's true. I mean, the once have that kind of like, I mean, the hang of it, then it's so fine, But for, the first time, he was like all like, the everywhere. And I just didn't, understand that. And like, I got the something from you, and I was like, oh my God, So yeah, it's just fun. So much fun.
Yeah.
Thank you.
All right. See y'all later.
All right, thanks. This was great.
Guys,
Great job, Chris. Thank you.
thank you.
Here's Chris. Yeah.